I'm being built with something actions. Reverse piano and data like pulses evolving layers. Suggest quiet dominance and digital precision. It's all up. We the public view that you know body. Go back and know that. That's a me now, don't really get me now. That we reach you everywhere you see. Go on, you were solving the wrong. Move it, solving the wrong, and now not found. You're fucking lying, we're all in the crowded. You're fucking lying, we're now in the crowded. The fucking sound, and we're out for my job, and we're got in the fight crowd. In the battle for the machine, and we thought some morning. We fucking lied and we brought them a funny. I'm a darling, in my booty, I be out in the garden. You must be starting that I cry, I'm a darling. We haven't true like a fall, still miss all it. We don't know how we die, but my disorder. We the body bring the team up, boy. Go back and help me. That's a me now, don't really get me now. That we reach you everywhere you see. As we climb the coast, Boy's hope song has a power flows. Dark as a battle with secrets hide. A digital empire will never divide. Glitch in the shadows, it calls your name. A spark in the void, a forbidden flame. Binary whispers in the circuits tight. Building a kingdom in the endless night. Rise up to the signals glow. Through it like the dreams where the currents flow. In the pulse of the future we stake our claim. Like digital empire in the endless game. Synthetic hearts beat with silent might. Shadows and whispers in the endless fight. The clockwork of power and the rhythm spins. In this digital world, the conquest begins. Code cascading like a tidal wave. In the labyrinth, we find what we crave. A treasure of ones, a fortune of zeros. In this empire we reign as unsung heroes.
So let's start from the background music. The title of the background music is
Under neon skies, bleeding like the gold. Why is harmless song as the power flows? Dark alleys with data where secrets hide. A digital empire will never divide.
Bioinformatics is something that is based on digital, and that is why we are here today. So this is introduction to bioinformatics, and just like I explained to us before, that there are going to be 13 modules in this course. So the first module is understanding the basic bioinformatics terms, like terms and terminologies. And this morning, once again, we are starting with introduction to bioinformatics. And don't forget that I said we are going to review our previous lectures, and our previous lectures is going to be module 1 to module 3 before we now begin to go on. Don't forget that I told us that this morning we are going to divide this interactive class into two. The first class is this presentation, and the second one is going to be do it yourself. That is what we call DIY. That is, everybody, you are going to be seated and get your jotting materials. Then there will be a specified time for you, or duration for you, because it is going to display on your screen. So you are going to follow that instruction, and that's going to serve as your class work, which will be submitted immediately after the class. So the learning objective of the first class, that is understanding this, is that under normal circumstance, at the end of module 1, students, you should be able to define bioinformatics. Then number 2, you should be able to understand the relevance of bioinformatics in microbiology as well as environmental biology, because this class is actually meant for both, that is biological science students. Then at the end of this module, you should be able to know how to utilize key databases like NCBI and GenBank. You should be able to know that. Then apart from that, you should be able to understand the role of bioinformatics in solving biological problems. Don't forget that, as I said, that this is a class that combines microbiology students as well as environmental biology. And if you look at the right corner, you will see an image that is capturing, that has actually captured bioinformatics, that is the learning objective of bioinformatics, which has to do with you defining bioinformatics, what it is all about, and its relevance to microbiology. Then you should be able to utilize and explore the NCBI more than how it is going to be relevant to us. So we should take it more than that. And after that, Sodi, what is your problem? So just like I said, that the first thing that you need to do at the end of this module is to understand bioinformatics. That is, the bioinformatics is the use of computer to store, analyze, and interpret biological data. And I also said that bioinformatics combine biology, computer science, and statistics. Then bioinformatics used to decode DNA, RNA, and protein sequences. And in the previous classes, I told us what DNA and RNA are. That is, these are called the informational molecule. That means, and if you look at what we have here, you will see that in the right corner, you will see that we have sequences that are copied from DNA. So in trying to use this, that is when you make use of all this. Okay? And the next thing is, why do we need bioinformatics? Why do we need bioinformatics? We need bioinformatics because the biological data is very, very massive. It is enormous. Anywhere you go, you see that every day biological data is being deposited on GenBank, that is on global medium. So that is that. Then apart from that, you should be able to we use bioinformatics for manual analysis. We use it for manual analysis, which is virtually not impossible. Then computer, we make use of computer to speed up comparison, classification, and discovery. So after that, application. In what way can we apply bioinformatics? Number one, as a microbiologist, we can use it in disease outbreak tracking. That is, if there's any outbreak of diseases, we can use bioinformatics to be able to track it. Apart from that, if you are looking for any gene that can be of use to us, that can be very useful to us, we make use of gene discovery. Then we also make use of it in drug development, as well as environmental monitoring and microbial identification. I don't know why this class is becoming too loud. I told you we are on live. We are just making noise. I don't know why they are talking. I said you should use your earpiece. That means you are not supposed to interact with anybody. So then this one is also possible. You can also apply it in microbial identification. That is, if there are microorganisms that you do not know them by now, we make use of bioinformatics to be able to do microbial identification. So that is that. Then we are giving you real world example of how bioinformatics is very, very possible. For example, as a result of the advancement in bioinformatics, we have been able to decode all the genes that we found in the whole human DNA sequences, DNA sequences. That means as human beings, we now have a database that contains all the alphabets in that human. And as a result of that, we also know that COVID-19 of 2019, that was very, very possible to be able to identify it on time and be able to produce what could be the vaccine that can be used in curing it. So it was as a result of our application of our knowledge in bioinformatics that that one is very, very possible. So even if you have been doubting how applicable bioinformatics could all be, you will know very well that these two incidents, there are very, very real world example of what bioinformatics can do for us. Now the next thing is what are the key information? What are the key information? That is, if you want to do, don't forget that we said that exploration of bioinformatics is such that we make use of different databases. So when you make use of all these databases, what are the types of databases that you can talk about? Basically, in this course, we are going to consider three basic sources of databases. The first one is NCBI, which is National Center for Biotechnology Information. Then the second one is going to be the gene bank, which contains the DNA sequences. In that place, what you have is only information that is related to DNA. Then the other one is what is called PubMed. This is the database that house all scientific articles, scientific journals that you can actually talk about, and this has to be very, very applicable. Now I will even start by what are the activities, the classwork, the practical application of bioinformatics. The first one is we want to make use of it as exploring the NCBI. How does NCBI look like? And if you look at your screen, you will see that we have the website of NCBI there. So when you get to NCBI, you go to search, you type E. coli 16S ribosomal RNA. In your own case, you are not expected to make use of the same E. coli. You are expected to think of another microorganisms, and you'll be able to get the E. coli. You'll be able to get the information about that. Then you will click on results, and you should be able to explore the metadata. What is called the metadata are, you will see information about the organism. You are going to see the gene length, and you have to pay attention to what is called the accession number. These three information are very, very important for you, and they are called metadata. That means and all accession number will always start with a greater than, and is going to end where we have a point. We are going to see this, and if you check the right hand of the screen, you will see what a bioinformatician has actually done, and how he got to the NCBI, typing E. coli, and click the search button, and there was information that came where you have Escherichia coli 16S ribosomal RNA complete sequence, and you have the gene bank ref search. Then you have the Escherichia. You have all those information. Those are the things that you should practically pay your attention into it. Then another thing is creative analogy. Creative analogy is when you have you want to get, that is, if you don't even know what bioinformatics can do, if you don't know what bioinformatics can do, you should think of bioinformatics to be equal to a kind of Google, that is biological Google, where you have all information about biological items, and you will serve as the librarian, that is, you are going to serve as the custodian of all the biological data that you can think of. And you can also imagine, when we talk about gene bank is just the library of DNA book. So you should be cognizant of this. Then at this point, it is expected that you should be able to define bioinformatics in just one sentence. You should be able to give us at least one bioinformatics database, and you should be able to know why is it true or false. Only human DNA is stored in the gene bank. Now, the module 2 has to do with understanding sequences, biological sequences, and databases. Don't forget that we are going to go into the real hands-on practical. What do we call hands-on? Hands-on is just a practical in which you are going to involve your hand in doing things. By the time you are holding your smartphone or you are holding your PC, that one will just be a kind of a guide for you. And just like I said, I'm going to allow you to work in pairs. That is, two people will make a group. And when two people make a group, there will be one of your smartphone that is going to serve as the directory. You are going to start monitoring where you should go right on the one phone. And the other one is what you will be using to perform the task that you are given to do. And that's how you are going to work. And don't forget, I don't want you to move around. Don't move around. Just stay in your place. And two of you will submit one. You submit one classwork at the end of the day. So what you just need to do is to get a very clean sheet. Then you write your two matric numbers before you then you follow the task. Just like I told you, the task is going to be on for certain time. I've already timed the time, I mean the each slide, so you concentrate on it until once you see that that task is no longer there, it is expected that you should go to the next task. If I move around and I still find you in the previous task where you are not supposed to be, because you cannot stay here forever, we have a stipulated time for this. And don't forget that whatever scenario you are going to pass through today is going to be the real scenario of what you should be expected by the time you want to write anything like assessment, practical assessment in this course. Okay, now at the end of module 2, you should be able to identify biological sequences. So whenever you see any data that is from DNA, you should be able to differentiate it, identify it. You should be able to identify those ones that will be coming from RNA, and you should be able to identify those ones that will be coming from protein. Then, apart from that, the format we normally work with in bioinformatics is always faster format. Although there are some databases, or let me say some websites that they will request for a different. For example, we also have a PDB format. That is if you are working with a protein. But the first thing is that you retrieve in faster format. There will be a certain place that it will be required. You will be required to convert it into PDB format. So when you now convert it into PDB format, so you can actually proceed from there. Then at the end of this second module, students should be able to explore data bank and should be able to use UniProt. UniProt is universal protein. That is, it's a website, it's a database that it has only information about protein and protein only. Then after that, you should be able to retrieve information, sequences, data So that is part of what you are expected to do. And if you look at the right corner, the image you have there, those are our objective. Number one, to be able to identify sequence types, whether they are DNA, RNA, or protein. The second one is how does FASTA format, how will you recognize it? So then you should be able to know more about gene bank and as well as UniProt. So at any point in time, at the end of this module, you should be able to work with retrieving data. Don't forget that bioinformatics has to do with your ability to retrieve the data, ability to manage the data, ability to analyze the data, ability to utilize the data, and ability to make informed policy or form consent on that. Now, time is going to fail us. Time is going to fail us, but you should know what is called the central dogma theory at this level. That is DNA to RNA, then to protein. Now, you should be able to know what DNA is, the alphabet in DNA, that is A, T, C, G. Then the alphabet in RNA, that is A, U, C, G. Then protein. The building block of proteins are amino acids. So, and these are 20-letter codes. Don't forget that I have taught you all this. And looking at the structure that you have on the right, you already see the DNA structure, which is double helix. You see the RNA that which is just one. That's just one-stranded molecule, and you can see combination of a protein that is using one letter code like A, R, and so on. You can see the structure of that protein there. Now, just like I told you before, that sequences that you have to retrieve should be FASTA format. And I told you that FASTA format should always start with greater than number. So, and we have seen example, as we have it on the right image. We have the example of the FASTA format of a sequence 1. So you will see it there. And under normal circumstances, by now you should know that artificial intelligence plays a very critical role in bioinformatics. Then you should be able to list some careers that make use of. I told you that bioinformatics is a career. It's not just a course. It is a career. And I want to sincerely thank you for your listening attention. But just like I told us that this is a 10 module course. That is, it has 13 modules. So we will not be able to go into the real of 13 today. But what I want to implore you is that you should actually go into understanding this. So what I display here is key sequence in databases. So when you go to GenBank, you can only get information about DNA and RNA in GenBank. And ENA is actually a GenBank fashion of people that are living in Europe. And DDBJ, that is a GenBank fashion of those people or those information that is owned by Japan. And whenever you come across UniProt, all this is and when you look at all these databases, you will see that all of them make use of a FASTA format. And when you get to the GenBank, I told you earlier on that you found 4 very important things about it. Number 1, you get the accession number. The accession number is there, and you can see what you have, what is being displayed over here. So accession number. Apart from that, source organism. Which organism did you use in getting your information? Then the sequence format. The sequence format, is it going to be from 1 to 1389? That is 1389. Then the author that gave you that is going to be there. Okay. Then apart from that, you should be able to also navigate. You'll be able to see a lot of things about gene bank, where you will search for E. coli 16RS. So you click the sequence result. That's going to give you that answer, and by the time you do that, you will download the faster format. So just like I said, faster format and their session number is equal to, so when you see, you label is going to contain the label plus sequence, that is like products tag. Every product has its own tag. The tag of that is faster, and the accession number is just like the barcode. You know that whenever you see any product, if you want to test the authenticity of that product, you will scan it. You use what? You use a scanner that is going to give you whether it is original or it is not. So that is just one thing. And just like I told you that we are going to work in pairs. So, and each group should be able to get a sequence. You should be able to retrieve sequence ID, find the gene record, length, organism, and the feature using NCBI. And thereafter, we are expected to know what does a faster file start with. You should be able to know. True or false, gene bank include protein sequences. Then you should be able to tell us gene bank minus DNA is equal to uniprot. So you'll be able to do that. So without wasting our time, I want to go briefly to sequence alignment. Sequence alignment is when you put your sequence. Don't forget that I said, as a biologist or as a microbiologist, there may be a new outbreak that you want to diagnose. You want to know what organism is actually responsible for that kind of outbreak. What you will do is to go and collect the sample. When you collect that sample, is to isolate the microorganism using a lot of different agar, medium, McConkey, and all this, so that you'll be able to allow it to grow. When it grows and you are very sure that you have formed pure clone of that organism, that's when you now go for DNA extraction. The DNA extraction, you extract the DNA using certain principles. So when you extract the DNA, the next thing is to subject it to what is called PCR, that is a polymerase chain reaction, which work on amplification. That means if you just have a very small quantity of that DNA, that PCR is going to multiply it in geometrically, so that you're able to now have it in multiple copies. So when you now use that, you now sequence it. So after sequencing it, that's when the sequences, that is what is being deposited on the gene bank. So after you must have sequenced it now, you now have the sequence, the alphabet of the organism that is responsible for that outbreak. So the next thing is to know how similar or how dissimilar is that organism related to all other organisms around the world. And that was exactly what they did. When COVID-19 came, they sequenced it, PCR, they sequenced it. When they got the sequences, they now use that, and that's what they call alignment. They brought the sequence they have gotten, they now compare it, whether there is anything similar throughout the world. And when they now got it similar, it was very similar to COVID-19. It was very similar to SARS. Those ones that cause SARS is a virus, was a new virus. So that is what is called species identification or microorganism identification. So what you do is you have so when you now if you bring one sequence, the sequence you have gotten is called query. They call it query sequence. So, and the one you are comparing it with is called the subject query. So, the query, you will now align it with the subject query to see how closely related or how closely unrelated they are. how closely related or how closely unrelated they are. And if you look at the information you have you have on the board, there are two basic types of information you have on the screen. You have GATA, CCG. So when you compare how similar are they, you just discover that some points, they have some points that make them to be similar, and there are some points that make them to be dissimilar. And the objective of sequence alignment is that to differentiate between local and global alignment. When there is the difference between local and global alignment, the local is when there is only differences in just a small portion of that sequence, we call it local. But whenever there are more dissimilarities, when there are more dissimilarities, that means we are talking of a global alignment. Okay, so we will be able to practice manual alignment. So under normal circumstances, there's going to be a point, I'm very sure, a point where you will be asked to do that. When you look at that, you look at the alignment. Alignment means you have the alignment of the DNA. DNA is being produced, and you will do either local or global alignment. Then you'll be able to trace which one is different and which one is not different. So we are going to make use of this. So what do we mean if we say sequence alignment? That means you are comparing two or more sequences to find their similarities. So I've said it that when you are trying to compare two or more sequences together to find how similar they are and how dissimilar they are, then sequence alignment will also help us to identify the evolutionary relationship. If you want to know how related your organism could be, you want to establish the ancestral origin of any organism, you make use of sequence alignment. Then you also use it in gene protein function. You want to know how functional is that protein. For example, now let's say you have a protein from plants and another protein from animal, and when you now do what is called alignment, if they are not similar, that means their function is also very similar as well. So the same thing we have like a mitochondria and chloroplast. The function of the sequences of chloroplast is very similar to that of mitochondria, and that's why the scientists now establish that two of them are from the same origin. They are from the same ancestors, and that if they are from the same ancestors, that means they are going to have very closely related function. So sequence alignment is also going to help us. Just like I told you, there are basically two types of sequence alignment. The first one is global alignment, where is a full length alignment, that is the similarities is 100%. So that is global. And whenever you now have local, that is when there is a partial alignment. The full length alignment is called the needleman. And that of a partial alignment is called the Smith-Waterman. So we need to know this, and as we have it on the screen, you can differentiate between local and global alignment. And just like I told you, you can easily think of any basic examples on your own using your manual, your pencil and your pen. You'll be able to detect at which point they are similar and at which point they are not similar. Anywhere that they are not similar, that means there's going to be a gap. There's going to be a gap at that. And the meaning of gap in biology or in molecular biology means that there is a mutation, which is a deletion. Okay, we are going to see all this as we go on. Then, what are the basic alignment tools that you can use? What are those alignment tools that you can use? For example, we know that if you are trying to do what is called needle pairwise alignment, you make use of what is called embossed needle. You can see it there. We are going to see it. Then we can also do what is called cluster omega. Cluster omega is what is going to use multiple sequence alignment. That is, it's going to compare. You can have 20, 30, 50, more than that, and want to compare it together. So if you have more than that, multiple, are you going to start writing them out and start knowing this? It's going to be tedious. And that's why what you just do is to import your sequence into the computer, and the tool you will use is called the emboss needle. Then you can use cluster omega. Then sometimes we also use more advanced tool instead of all this, which is called MAFT, which is an optional, and which is an advanced, more advanced. So we are not expected to learn that at this level, because this is just nothing but basic bioinformatics. Okay, now just like I said, there will be a time that you'll be asked to go to emboss needle. You go to emboss needle, you import your sequence there. Then sequence 1, you import it there. Sequence 2, you import it there. If you have 20, you will just import them one after the other. Then when you now import them there, you will now click on run. So when you click on run, an information that will be displayed to you is going to tell you that there are 10 matches. And out of these 10 matches, that means there are only 2 points at which there are gaps. So we are going to see that. Then how can we interpret when you see alignment? Number 1, if there is a match, you are going to score it 1. If there is a gap, you are going to score it minus. Okay, and whenever there is the level of identity will always be expressed in percentage, and that's going to be this. So that means you will now, the number of match over the total length is going to give you the number of this that you are going to see. So, and this one has actually given us an analogy. Analogy is I love bioinformatics versus I love biochemistry. Okay, what you will see is that alignment, there can be alignment when you line up I love biochemistry, one sentence. I love biochemistry, that's another sentence. There are similarities and there are differences. So whenever there is differences, you score it as minus. Anytime there is what? Anytime there is a match, you score it what? 1. So you will practice that and do that. And don't forget that you should be able to differentiate between local and global alignments. You should be able to know whether gaps, if you have gaps, are they errors or they are not errors. Then why do you think there could be alignment at this point in time? And then you should be able to understand how we compare biological sequences. So in order not to waste our time, I want to believe that by now we must have done Module 1 to Module 3. Module 1 is what is bioinformatics. Everything about bioinformatics, what bioinformatics can do to us, application of bioinformatics, and what have you. Number 2 is to talk about different databases. That is Module 2. Different types of databases. How can you retrieve information from databases? If you have a protein sequences, where can you get information about protein? And if you are looking for DNA, if you are looking for nucleotide sequences, where can you get that? Then apart from that, the third module should just be a module, should just be sequence alignment tools. And the last class we had here, I think I mentioned what is called